About seed collection with particular reference to how we use those collections for translocation. And I think one of the main messages I'd like to say in relation to seed collection is start early. And when I say start early, I'm not talking about months or whatever, I'm talking about years. And even if it was something that we were collecting to translocate straight away, you probably need to be thinking in the order of a couple of years. And beyond that, I'm going to show some examples of why, in reality, you need to be sort of thinking at least 5 years in advance. So first off, for a general seed collection, how much seed are we aiming to collect? Well, the guidelines indicate we should probably be looking in the order of between 3,000 and 20,000 seeds. And this quantity of seeds hopefully gives enough to be able to test the quality of those collections, to monitor the viability or the quality of that collection through time, to duplicate it, so all the seeds not in one place, it's in safekeeping elsewhere, and then also to be able to use those collections. So just for a collection, we're talking thousands. But how much seed do we need if we want to undertake a translocation? So in this figure here, we can see the results for 4 species which have been translocated here in Western Australia. And at each step along that those lines, it shows the losses. So we start with our seed, and then we try and germinate that seed. So not all of those seeds are going to germinate. So we've already sort of reduced a bit. Those seeds then grow on in the nursery. Not all of the plants survive through the nursery. So again, there's further losses. And then the final step is actually when those plants go out into the wild. And again, there's losses at that step of the process. So if we want to establish a population in the order of 250 translocated plants, this work's showing us that for one of the better examples, we would need in the order of about 900 seeds to establish those 250 plants. And at the other end of the spectrum, for one of the pea flowers, we need close to 8,000 seeds. And just on average across those 4 species, it was about 1,800 seeds. So again, we're talking of needing thousands of seeds to get a relatively small number of plants. And if we're going to use our seed collections for translocation, we need to remember there's no guarantee of success. So when we're using that seed, we always want to keep some in reserve, just in case our translocation doesn't work. We've got some material to come back to in future to use again. And this is particularly important if we've got collections from either populations that might have become extinct or populations have been through a big decline in plant numbers. So if we wanted to go and recollect those seeds, it might not be possible now. So our ex situ collections represent diversity that may not be able to be replaced. And so to put these numbers into context, here at the West Australian Seed Centre, if we look at the size of our collections of threatened species, we can see that 3/4 of our collections are falling below the level of about 1,000 seeds. So most of those collections are very small. That's just based on a one-off collection, and one collection doesn't do everything, and often we have multiple collections of a species. So if we bring that data for all of these species together, so for most of our collections we've made multiple collections. On average we've got about 8 collections of each of our threatened species, but the numbers range from a single collection right up to one of them it's over 70 collections. But even then we can see, adding all these collections together, still 1/3 of those are less than that 1,000 seed, and only 1/3 of them are above sort of that 10,000 seed, which is actually getting into the level of what we might really require to be able to establish our plants, have some in safekeeping. So what do we do in these instances where we just can't get enough from the wild? And that's where we need to consider things like seed production areas. So plants grown in a managed situation where hopefully we can improve the survival of those plants, and hopefully we can increase the seed production of the plants that are grown there, or at the very least we can harvest all of the seed that's produced, as opposed to sticking to safe collecting guidelines, which is what I forgot to mention when we talk about making our collections. When we're collecting from the wild, we want to make sure that we're not impacting on those populations. So there's limits to how much seed we should be taking at any one time. And the guidelines indicate, the upper limit should be about 20% of the seed in a population, but that's based on a once-off collection. And as I've shown, for a lot of these species, we may need to be going back numerous times, so we need to be far more conservative than that. So for a lot of species, the idea of a seed production area needs to be considered. So when we're going to go and make our seed collection, it's obviously important to make sure we collect what we want to collect, the species we're interested in. And there's a whole range of resources we can use for that, from botanical descriptions, scans of specimens and those sorts of things. So that's when we go out, we collect, make our collection, and hopefully we've got the right thing. But it's vitally important to make sure when we make that collection that we take a herbarium specimen to vouch for what we've collected. So in the first instance, it makes sure that we've got it right. What we've collected is what we thought it is. But also of importance is it future-proofs the identity of that seed collection. And in the example I've got here, I don't know whether you can see that, but this is a West Australian genus called Gelesnauia. And when this specimen was taken, it was considered to be one species. More recently, taxonomic and genetic work's been done on this species, and what was considered to be one species is now 7. Okay, so, and some of these are co-occurring. So I know of sites where you can get 3 of these species all co-occurring. So without that herbarium specimen, down the track, you may not actually know what actual species you've got. So it's important for future proofing to make sure you've got the right thing. Diversity is everything when it comes to seed collections and for using those collections for translocation. So when we make our collections, we want to make sure that we sample adequately, not only within a population, but also between populations or across the distribution of the species. We say we aim to collect 95% of the variability within a population, and the general rule we would use is if you've got at least 50 plants in a population, you collect from at least 50 plants, or if your population is smaller than that, which is often the case for our threatened species, you try and collect from as many of those plants as you can. We want to make sure we're not biasing our collection. We don't just go for the big plants or the pretty plants or those sorts of things. We want to represent the diversity. So we need to, if there's a large enough population, randomly collect from across it. Ideally, when we make these collections, we keep each individual plant separate, and that value adds to our collection. So it may allow for genetic studies in the future, or when you're using that seed for translocation, you can follow maternal lines to see what the diversity is in your translocated population. And as I've said, we need to make sure we collect across the species range. So I'm going to give you a couple of examples now of the importance of collecting across the range of a species. So here we have an example for one of our critically endangered species, Banksia brownii. Now, I'm not sure if you can make it out, but the black squares represent currently extant populations. And I should say this is a species that we have in our collection dating back to the late 80s, and it's been collected from then pretty much up until the current time. So the black squares are currently extant populations, but we've got some diamonds in there, and they're surrounded by some circles. They're actually populations that are now extinct, but are represented in the ex situ seed collection. And so a genetic study was done looking at the diversity in our seed collections, and it was finding that there's more diversity in the ex situ collections than can be collected and replaced in the wild at the moment. The other thing that the study showed was that we actually have three conservation units for this species. So the ones which are surrounded by the purple circles up there is in the Stirling Ranges. There's another band of populations through the middle, and then there's a final conservation unit down on the south coast. So another example of the importance of sampling across the populations is this is a species that was known as Stollidium coroniforme. And when the seed centre was first established, this species was known from two clusters of populations, about 100 kilometres apart. So seeds were collected from both of those clusters to make sure that the species was represented. But more recently, it's been found that those two clusters, which at the time were thought to be one species, are actually two completely different species. And you might notice there's some more dots down the bottom there. In the meantime as well, other populations were found, which was found to be a subspecies of the Stollidium coroniforme. So, and those have now been conserved. So again, if we had just taken the single population approach and we had focused on maybe the Wongan Hills population, a completely different species wouldn't have even been represented in the collections. But I guess the flip side to what we're seeing here is the considerations that you need to think about when you're using collections and which populations you might mix when you come to use those collections for translocation. Because if by chance you happened to, in the original days, if you'd mixed those two things, you would have actually been mixing two species together in a single translocation. So it highlights the importance of thinking about population source when you're undertaking your translocation. If we're talking about getting in early, we obviously want to make sure that that seed is viable when we come to want to use it. So we need to store the seeds under conditions that prolong the longevity of those seeds. And for most species, storing the seed at low moisture content and freezing the seed below minus 18 degrees are the conditions that's recommended for most species. When the collections go into storage, we want to know what the viability of those collections is. So that tells us, from a practical standpoint, the utility of our collections. How much of our seed are we going to be able to generate plants from? But also it gives us a point from which we can monitor the viability of that collection over time. So that viability testing needs to be done up front, but then periodically through time to make sure that that viability is being maintained. And at some point in the future when we come to use that seed, that it's actually going to be viable. Finally, good record keeping is vitally important to successful translocation. So I guess in the ideal world, with our collections, plants might be tagged in the field. So a wild plant might have a unique number. We'll collect from those plants, and when it comes into the seed bank, we then give those collections a unique identifying number, so we know where those collections have come from. And then that good record keeping means that we can track that information through the whole process. So when the plants go to the nursery, records are kept about where that seed has originated from, and we can trace it back to a plant in the wild. And then again, when those plants are planted into our translocation, again, those plants are tagged so that ultimately, in your translocated population, in the ideal situation, you can trace it back from your translocated plant to a wild plant out in the wild. And then you can monitor how maternal lines and those sorts of things are going. Thank you.